Hello everyone, good afternoon. So we're back for sex-linked trait inheritance. So we're going to start with a definition of sex-linked traits. So these are any trait that is controlled by genes that are found not on your autosomes, not on chromosome 1 to 22, but on your sex chromosomes. We know that the 23rd pair, it's the X and the Y, those are your sex chromosomes. So these particular genes are not found on chromosome 1 or 2 or 3 or 5, they're found just on the X or on the Y. And so because of that, because we find them on our sex chromosomes, what ends up happening is that these traits will occur more often in one gender or the other. Okay. And to explain that, I'm going to use X-linked and Y-linked genes. So an X-linked gene, also known as, you're going to hear it as sex-linked. These are the ones that they call sex-linked on questions or in the textbook. So these particular X-linked genes, they are found obviously on the X chromosome. And you would think that they would be in higher frequency in females. You would think that because girls have 2 X chromosomes, that you would see these traits more in females than you would in males. But it's actually the opposite. You end up having X-linked traits will occur more often in males. Why is that? It's because males only have 1 X chromosome. Females have 2. So if you have a recessive condition that is passed on the X chromosome, males will only have 1 X, and therefore they will inherit that recessive allele. Females have 2 X's, so even if they inherit that recessive condition on one of their X chromosomes, they still have another one that could inherit the dominant condition and therefore cancel out the recessive one. Okay, and we'll see an example of that in a cross in just a second. Okay, so these are the ones that occur more often in males. What are some examples of X-linked conditions here? So we have a few. So examples include myopia, so being nearsighted. So if you need glasses to see far away, you can see fine near, but you can't see well far. That's called myopia. That's an X-linked condition. It's found on the X chromosome. And also we have red-green color blindness. So this is a deficiency in the cones in the eye that detect red and that detect green. And so those are the 2 deficiencies that you're going to have, the red and the green color blindness, and also hemophilia. These are the ones that come up most often. Hemophilia is the inability for the blood to clot. You are missing one of the clotting factors if you have hemophilia, and therefore if you start to bleed, you're not going to be able to clot and make a scab and close the wound off. So that's the problem with hemophilia. Okay, so these are just some examples. And then you can have Y-linked, also known as Holandric. And these are found exclusively on the Y chromosome. And again, it seems that here the males are the ones that are getting the short end of the stick because these ones don't occur more often in males, they occur exclusively in males. Okay. So we said those ones occur most often in males, and here we said it's because they only have 1 X chromosome. And the Holandric occur exclusively in males because they are the only ones that have the Y chromosome. And again, we have a number of examples that we could use for Y-linked conditions. There's, they're not as common as X-linked, but there is a few. One of the ones that you might find interesting for Y-linked is something called hypertrichosis. And it's hypertrichosis of the pinna. The pinna is this part of the ear right here. And so men are the only ones that can have really, really long ear hair because it's part of the Y chromosome. Females don't have that particular condition available to them in their genome. Yay, good for us. Okay, so this is extremely long. Oops. Ear hair. Oh. Okay. So that's an example of a Hollandric condition. So who do we have to thank for the discovery of X-linked traits? Again, it is Thomas Morgan with his work in with fruit flies. And so before we move on and look at some crosses, let's have a look at Morgan at the fruit fly. And I'm going to put in brackets again because we saw that with multiple alleles. And so he studied eye color in fruit flies. We saw that those eye colors could be red and apricot and honey and white, but he also noticed that when he crossed red-eyed flies with one another, a lot of the times he would get red eyes, and makes sense because it is the most dominant of the colors for red eyes. And then once in a while he would keep crossing red-eyed flies with each other, and once in a while in the offspring he would spot a white-eyed fly. So once in a while you would have two recessive genes that would come together to make a white fly. But the thing that Morgan noticed is anytime that he got a white-eyed fly, it was always a male. And so he started to thinking there has to be a connection between gender and this particular eye color. So let me sort of summarize that for you. So when he crossed two red-eyed flies, occasionally he would get a white-eyed offspring. But it's not what interested him, like I said, but that offspring was always male. So there had to be a connection. There had to be something to connect the two, okay? So what did he propose? He proposed that the gene that controls eye color in flies, not in humans. In humans, interesting enough, there's 13 different genes that control your eye color. So we tend to simplify and say brown versus blue, but it's actually a lot more complicated than that. So the gene for eye color in flies is found on the X chromosome. All right, so let's see, given this information, then what did Morgan do when it came to alleles and writing crosses for Punnett squares? So he said, okay, I'm not going to be dealing with just dominant, recessive, incomplete dominance, codominance. I'm not going to be dealing with that. I have to deal with the X and Y chromosome because gender has something to do with this. So what he said is I'm going to have an X chromosome. I'm going to have a Y chromosome. Now for the X chromosome, I have two possibilities. I can have the red eyes, or I could have the white eyes. The Y chromosome doesn't carry that particular gene. So the Y chromosome doesn't get a superscript, okay? It stays blank because the only thing that is going to be carrying that allele is going to be the X chromosome, okay? So in this case, then we can say this is red, and this one right here is white, okay? This right here, it just gives me male. It just makes it so that the fly is male. So in his crosses, if I had to do a Punnett square, he crossed 2 red-eyed flies. So I'm gonna need a female and I'm gonna need a male fly. So that's where I put the X's and the Y. And one of the flies, the female has to have red eyes, so I'm gonna put red. I'm not gonna put the other one because I don't know what the other one is. All I need is one of those big R's because the condition is dominant for me to have red eyes. The male also has red eyes, so he has to have big R. And here, nothing, because the Y chromosome doesn't carry that particular gene. It doesn't have it, okay? So then when he had the cross, he had females and he had males that came out of that cross. And you can see that by doing X with X, X with X, X with Y, and X with Y. Now you can see why the percentage or the probability of having a male or a female is 50%. When I cross a male and a female together, what I get is 50% are female, 50% are male. Okay, so I get an X and an X or an X and a Y. That's why the percentage is the way it is. Also because there's only 2 genders, but that's why you have it that way genetically anyways. All right, so then let's look at what happens with our letters, okay? Because we do have our X's and Y's, but what about our superscripts? So here I have a red-eyed female. Here I have a red-eyed female. I'm just gonna leave that other one blank. I know I have red. Here I have a red-eyed male. Now one of these offspring has to be white-eyed. And so that means that this particular letter right here needs to be a little R. And then there is my white-eyed male, okay? This individual got a little R from mom and the Y from dad because it's a male. So that's the only thing dad can give him, okay? So that's right here. We're gonna indicate this is the white-eyed male, okay? So that's what we get when we do the cross that Thomas Morgan was dealing with it. And so to sort of reconnect that to what we said earlier when it came to X-linked and Y-linked, why does it occur more often in males? Why does it occur exclusively in males for Hollandric? Then what we can say here is that females need 2 copies of a recessive allele to have the condition. Whereas males only need 1 recessive allele, since the Y does not carry the gene. Okay, so this is an X-linked, obviously. In Y, it would just be the Y that has the chromosome, that has the gene. It wouldn't be part of the X. And we're not going to be doing any crosses with Y. I just need you to know that they do exist. Okay? Now, the one thing that I need you to pay attention to, and it's something that happens every time, and that's why I'm make sure that I write this down. Attention. Okay? Because what tends to happen is after I teach X-linked, you guys want to make everything X-linked. As soon as I say a man and a woman mate to produce this, you automatically want to put X's and Y's up in your Punnett square. And as we've seen so far with dominant recessive, with incomplete dominance and codominance, and with multiple alleles with blood groups, we've always had a male and a female mating together. That's how we make gametes. That's how the gametes come together to make zygotes. So the part that you need to pay attention here is that there are only sex-linked conditions if these parameters are met, okay? So only sex-linked if it says so in the question, okay? So if it says so in the question, in the sense of you might have something like this. This is a sex-linked condition. Okay. or it might say something to the extent of this gene is only found on the X chromosome. Okay. So only if it says so in the question, or those conditions that I told you earlier, those ones, they're sort of, if you were writing a diploma exam, they would sort of think that they would want you to remember those conditions. So if you're dealing with myopia, if you're dealing with red-green color blindness, and if you're dealing with hemophilia, these guys, you need to remember that they are sex-linked conditions. So it might not say that it's on the X chromosome, it might just talk about hemophilia, and that you need to know that it is an X-linked condition, okay? So let's look at another problem. I'm gonna give you a question right now just to see if we can do it together. So we have color blindness. Color blindness is found on the X chromosome, and it is recessive. A woman with normal vision whose father was colorblind marries a man who's colorblind. And it wants two parts to this question. What are the chances of having a normal vision girl? And then what percentage of their boys should be colorblind? So we have to, approach it like any other question that we have so far. We're gonna start by sort of writing down the genotypes of our parents and maybe getting them onto a Punnett square, okay? So let's see. A woman with normal vision. So she has normal vision. So I'm gonna just give it N for normal. And I don't know what the other one is until it says that her father was colorblind, okay? So if her father was colorblind, the only thing that we can have him have, he's gonna have the little N and the Y, okay? So he was colorblind, okay? So the only thing dad can give her when I look at his genotype, he's not going to give her a Y because she's a girl, so she can't get the Y. So the only thing dad can give her is a little N. So she got the big N from mom, and dad is giving her the little N. Now she marries a man who is colorblind. So he is colorblind, so he needs to have a little N and the Y because he's a male. We're going to do a Punnett square with that, and we get the two girls and the two boys. Big N, little n, two little n's there, a big N there, and a little N there. So now the first question says, what are the chances of having a normal vision girl? So what they're saying is, let's say they have a kid, what are the chances that that kid will be a girl and that it'll have normal vision? So here I'm dealing with a product rule. So what are the chances of me having a girl? So to have a girl, I need to have 50% chance of that. And then when I'm trying to figure out normal vision girl, what I'm going to do is I'm not going to be looking at the boys. I'm only concerned with the girls. So when I look at my girls, I have a 50-50 chance that that girl could be normal. So when I'm writing the probability for normal vision, what that probability is a half. So again, I multiply the top and I multiply the bottom. So I have a 25% chance that when I have a baby, it'll be a girl and it'll have normal vision. Whereas in the second question, what it's asking is, what is the percentage that their boys will be colorblind? So this question is taken into consideration that they've already had boys. I'm not saying what are the chances that they will have a boy. They've had boys. What are the chances that those boys will be colorblind? So again, what I'm going to do is I'm only going to be looking at the boys in this case. I'm not taking into consideration gender and what the probability of that will be. And when it comes to boys, I want to know colorblind is 50% the chance that I will have a colorblind child that is a boy. So that's 50% or a half if we want to write it as a fraction. Okay, so 50% or a half. Now, another note that I want to give you before we go into the next problem is the fact that you can have some terminology that sort of comes up with this. And so some of the terminology that you want to note is that a heterozygous female may be referred to as a carrier. Okay, so you might see the word carrier there, and you might have to right away think, okay, carrier, that means heterozygous. I have an X with a dominant and an X with a recessive. Okay, so let's see another problem here and how we would do this one. Okay, so we have this one right here. So we know that hemophilia is a sex-linked condition, and you have a hemophiliac woman who marries a normal clotting man. Their daughter then marries a hemophiliac man. Give the genotype and phenotype ratios for their children. Okay, remember that when you're doing a phenotype now, that's one of the notes that I'm making there, when you're doing the phenotype, part of your phenotype is your gender. It's not just what you look like, but it's also, because this is a sex-linked condition, part of your genotype is going to be your X's and Y's, and part of your phenotype, you have to write down the gender. Okay, so let's see. I have a woman who is hemophiliac. So she has hemophilia. So this is the parents I'm dealing with right now. So she's a hemophiliac woman, and I'm just going to use little H's, okay, for hemophilia. And she marries a normal clotting man. So she's being mated with a man who has normal clotting. And if you want, you can make a little legend. You can say X, big H is normal. X, little h is hemophiliac. Okay, so you can do that. All right, so they are going to have babies. So I'm going to do the first Punnett square. Little h, big H, Y. So here's my girls, one normal girl. Here I have a boy who's hemophiliac. Here I have a normal girl. And here I have a boy who's hemophiliac. Okay, so they're saying that their daughter marries a hemophiliac man. Okay, so their daughter, and the reason why they're saying their daughter is the fact that both girls are the same. So it doesn't matter. Anytime that they have a girl, that's the genotype that their girl is going to have. So I'm going to take one of these girls, and I'm just going to write down here, this is their daughter. And so she's big H, little h. She marries a hemophiliac man, and it wants to know the genotype and phenotype of their offspring. So X big H, X little h, little h, little h, big H, Y, and little h, Y. So now I have to do a genotype and a phenotype ratio, and I can sort of list them all. So I can say for my genotype, I can say I have that, and for the males I have this, and I have this last one. Okay, but now when it comes to the phenotype, I can say females, 1 normal to 1 hemophiliac, or I can say 1 normal girl, 1 hemophiliac girl. Or I can just say for the males, I have 1 normal and 1 hemophiliac. Okay. Sometimes what can happen, the last example I want to leave you with, is there is such a thing as X-linked dominant conditions. Right now we've been looking at X-linked recessive conditions. What happens if I have an X-linked dominant condition? They are rare, but they do exist. So the last question I'm going to leave you with is going to be one with an X dominant, and it's called Rett syndrome. So Rett syndrome is an X-linked dominant condition where you have underdevelopment of the brain, and you can have loss of movement and speech in the babies that are born. And attach another one. So then this is the question then. I have a woman with Rett syndrome, marries a man, and their children are half affected and half not. What is the genotype of the woman and the genotype of her husband? Okay, so again, I'm dealing with Rett syndrome. I'm going to be making a little key for myself. So I'm gonna say, okay, X big R is going to be Rett. X little r is going to be normal. Okay, so sometimes, so right now it's the little r that is the normal, and it's a big R that it is the condition. Okay, so a woman with Rett syndrome, so she has it. We don't know what the other one is. She marries a man. All we know about him is that it has an X and a Y. We don't know what the rest is. But what we do know is that half of their children are affected and half of them are not. Now, when I look at half of the children that are affected, this is the half that is going to be affected. This is the half that will get the X with the big R. So, X big R. So here is my girl that is going to be affected. And here's my boy that is going to be affected. Now the other half, this side, is not going to be affected. So because it's not, mom must be a carrier. She must be heterozygous. And so in this case, here we have X little r. Dad cannot have big R, because if he had big R, then he would have Rett syndrome, and he doesn't. Okay, so he must be little r. Okay, and we have that. And then here is the other half, and here is the half the children with Rett syndrome, and there's half the children without it. Okay, so that's the end of X-linked condition. So again, I'm going to post these notes up, and I'm going to post a worksheet for you guys to work on, and we will talk to you soon.